In this video on gene expression regulation or transcription regulation, you will learn everything you need to know about the arabinose operon, also known as the araOperon, which is a regulatory system in prokaryotes or bacteria. Specifically, in this video, we will discuss how the activity of arabinose operon is regulated in the presence or absence of arabinose. And our understanding will come through the mechanism of araC protein action, which regulates the operon both as an activator as well as a repressor. In case you don't know, arabinose is a monosaccharide. It is a pentose sugar, meaning that it has 5 carbons in its backbone. Arabinose is commonly found in instant coffee, wine, and even sake. Essentially, it is a plant sugar, but humans have a tough time absorbing or digesting this specific sugar. However, bacteria living in our gut, or just in general as well, can easily absorb or digest arabinose. The arabinose operon is the system in bacteria that helps in the digestion or absorption of arabinose. So this specific operon is a catabolic operon. So far, we've discussed the lac operon and tryptophan operon. The lac operon is an inducible type, and the trip operon is a repressible type. This categorization is based on the activation or the repressing protein involved in the mechanism. The araOperon is both an inducible and a repressible type of operon. And that is because the regulator protein in this operon can function both like a repressor as well as an activator. Let's start dissecting the arabinose operon. Since operons are transcriptional units on the DNA, let's understand the araOperon locus at the DNA level. The arabinose operon has 3 structural genes, called araD, araA, and araB. Collectively, they are referred to as araBAD. The araBAD is controlled by a promoter called pBAD, which has its canonical -10 and -35 elements. Just upstream of the promoters are 2 sites, which are direct repeats called I1 and I2. The I stands for inducible. And the I site is 17 bases in length, and the space between them is 4 bases. Upstream of the inducible site is a cap binding region, which, if you recall from the lac operon, is 20 base pairs in length. Upstream of the cap site are 2 sites. Again, these are also direct repeats, called O1L and O1R. The O stands for operator, L for left, R stands for right. Next to the operator is another promoter which goes in the opposite direction, which is responsible for transcribing the regulator protein called araC. In the coding region of araC is a single operator site called O2. All these operators are also 17 bases, just like the inducers. They are more or less similar in the sequence composition too. The most important thing to keep in mind is that the I2 site overlaps with the -35 element of the promoter of the araBAD, and the O1L overlaps with the -35 element of the promoter of araC. And these promoters are facing away from each other. Something that will be important later is that the distance between the I1 and the O2 region is around 200 bases. Anyways, let's get back to these sites here. You should notice that all these sites face in the same direction, and they're all 17 bases in length. Each 17 base site is called a half site. And if you have 2 of them together, well, half and half makes a full site. So the inducer sites and the operator 1 sites are full sites. But the O2 operator, which is in the araC, is a half site. So these are the regulatory features. And now let's talk about the structural genes in this operon. And because we have established that they are involved in the metabolism or digestion of the arabinose, they're catabolic in function. But what do these araBAD genes do specifically? We start with the arabinose and the araA gene, which encodes the arabinose isomerase, which converts the arabinose into ribulose. And then the ribulose is converted into ribulose 5-phosphate via the enzyme produced by araB, and this is called ribulokinase. Since we just attached a phosphate here to the ribulose, the reaction uses ATP as the source of phosphate. And then this ribulose 5-phosphate is converted into xylulose 5-phosphate. This reaction is catalyzed by the araD product, which encodes for epimerase. And these 3 are the araBAD genes. You may have come across this step of reaction in the pentose phosphate pathway as well. But anyways, this final product from the araOperon is converted into fructose 6-phosphate and glyceraldehyde 3-phosphate, both of which are substrate in the process of glycolysis, which can be used to make energy. So in this way, the araOperon can convert arabinose into usable energy. We have discussed the araBAD genes, but we also have this araC on the other end. And since we're discussing the genes, let's get into the araC as well, which is an arabinose binding protein. The araC has an affinity for the half sites, the 17 base pair site, at both inducer and the operators. There are 2 half sites at the inducer and the operator, and this one half site away upstream. The araC functions as a dimer. To draw a parallel here, we have seen the aporepressor of tryptophan operon, which is a dimer as well. This comes in contrast to the LacI inducer, which is functional as a tetramer. And likewise, these regulators are sensitive to their respective substrate that they regulate, which is tryptophan and the allolactose. Similar concept applies to the araC protein as well. But the araC is different in its function, because it functions as a repressor when there is no arabinose, and it functions as an activator when the arabinose is present. And this is what I also pointed out in the beginning when I discussed their dual function. Let's understand araC in a bit more detail and draw out the dimer. These are the 2 monomers, where the dimerization occurs at the N-terminal domain. The C-terminal domains face outwards. There's a linker region connecting the N and C-terminal domain. The N-terminal domain has this weird arm region that sticks out a bit. I will talk more about this when we actually get to its function later. This curvature in the N-terminal domain here represents the pocket at which the arabinose molecules can bind. And once again, to repeat, the heads of the N-terminal domain come together to form the dimer. This means that this affinity for half site comes from the C-terminal domain. The C-terminal domain of each monomer can bind the 17 base pair half site. A quick note on the half sites and the affinity: the I1 site at the inducer region has the most affinity for araC, and the second best site in terms of affinity is the right half site of the operator 1. Now we're ready to discuss the regulation of arabinose operon in terms of its arabinose consumption and operon activation. In case you haven't noticed, the arabinose is a sugar that comes from an external source, which begs the question, how does arabinose even get inside the bacteria? Especially if the araOperon itself does not include any transport proteins. Recall that in LacZYA, when we talked about LacOperon, there was this LacY, which was the transport protein called permease. In the case of arabinose operon, it turns out that the transport proteins are independent of the operon itself. There are 2 independent operons that control the transport of arabinose. The first one is called araE, which encodes for a single protein, which is a low-affinity hydrogen ion gradient-dependent importer. You may have heard of proton importers, and this is what this protein is. The second operon is the araF operon, which consists of 3 genes, araF, G, and H. This operon makes 3 proteins, which form high-affinity ATP-dependent importers. You may have heard of ABC family proteins, which stands for ATP-binding cassette family proteins, and that is what this transport protein is. As with LacY in the LacOperon, the araE and araF operon are always available at least at the basal level. Generally speaking, the regulatory structure of araE and araF operon looks very similar to the araBAD operon itself, where you have the same inducer and operator sites near the promoter. I will point out the regulation in context of arabinose later in this video. But for now, you should have a good sense of the basics, which is that the arabinose consumption depends on A, B, C, D, F, G, and H genes, where araBAD controls the catabolism, araC is the regulator, and the E, F, G, and H are the transport proteins of arabinose. Okay, now we're ready to dive into the nuts and bolts of the regulatory mechanism of the arabinose operon. I will quickly draw out the operon for clarity, and while this gets done, I want you to make sure that you have subscribed to the channel and have hit the bell icon for good luck. Okay, let's start with a case where there is no arabinose present for the bacteria to chew, which means that the araOperon must be repressed, which is another way of saying that the araBAD expression should be repressed. That's all good, but what about araC? Let's work with the simplest assumption that araC is present at least at a basal level. So that basal level of expression generates the araC monomer, which forms the dimer. This dimer can bind the inducer and the operator sites. If you recall, a few minutes ago, we said that the I1 site is the most preferred site for araC to bind. So let's sketch out how things may look when the araC dimer binds to the I1 site. Now, keep in mind, I am assuming that the default binding of araC is probabilistically higher at the I1 site. It is also possible that the araC binds to the second most preferred site, which is the operator O1R. But again, given all dynamics, the equilibrium that is established tips to the I1 site. So I will keep matters simple and choose the I1 site as the starting point. This other site of monomer is, however, free to bind DNA as well, right? It can target O1R, O1L, or even O2 site. Heck, it can even bind the I2 site, which is right next to the I1. So given all these options, where should the DNA binding occur? We need to understand a few things before we can answer that question. First is to go back on the idea of distance. The operator 1 and the inducer 1 site are about 50 or so bases apart from each other. The operator 2, on the other hand, is 210 bases upstream. The I2, on the other hand, is only 4 bases from I1. The I1 and I2 are simply too close, so the araC actually cannot bind I2. The araC dimer is actually not flexible. Now, there are two things that should pop out. One is the idea of being too close, which suggests a dependency on the distance. And then there is a concept of reach given the flexibility of the araC dimer. The reach of the dimer has a lower limit cutoff of about 80 to 100 bases. 4 bases means that I1 and I2 cannot be bound by the same dimer. The distance between I1 and O1 is less than 50 bases, which is under the threshold of 100 bases. So let's say that somehow it may occasionally try to bind O1L and O1R. But still, given that, the binding will really be unstable simply because the reach is under the threshold. The only option now available for the dimer is the operator 2, which is quite far away, well above the threshold. And that's where the dimer binds. Let's actually try to draw out how the binding of O2 would look like. Note that the O2 is facing this way, and the dimer is not flexible, which means that the dimer can only bind I1 and O2 at the same time if the I1 and O2 regions form a looped structure. If that happens, it'll look something like this, where the cap site and the operator 1 and the promoter of araC is caught inside this DNA loop. Although unlikely, it is possible that the RNA polymerase can bind the promoter of araC and initiate transcription. In such a case, the moving RNA polymerase will hit the O2 region, which is occupied by the dimer itself. So the RNA polymerase fails to move any further, and the araC is not expressed. Technically speaking, the loop causes the DNA to supercoil, which affects the topology of the DNA. And for our interest, this change in topology causes a steric block, such that the RNA polymerase cannot bind the pBAD. The pBAD is not in the loop, but it still gets affected because the distance between I1 and pBAD is only 25 bases. Promoter of araC, on the other hand, is caught in the loop, so the transcription of araBAD and araC is turned off. This is exactly what we thought would happen when you don't have arabinose in the cell. Similar mechanism also keeps the araE and araF operons turned off, but keep in mind that they are nonetheless maintained at a basal level. Now that we understand how the operon is turned off, let's try to understand what happens to the system when arabinose is present for the bacteria to consume. Naturally, you would expect that the arabinose operon is activated, which means that the araBAD expression is upregulated. This upregulation is driven by the change in the structure of the araC protein. Typically, araC monomer looks something like this, which has a N-terminal domain, a C-terminal domain, linker, and this arm. But when the arabinose is present in the bacteria, the arabinose can directly bind to the arabinose binding pocket, This binding causes a conformational change in the araC monomer. Following the conformational change, the monomer looks something like this, where the N-terminal domain arm flips out and acts like a lid for the arabinose binding pocket. In essence, this lid stabilizes the arabinose in the pocket. Okay, going back to this operon. As we just said a few moments ago, in the default non-active state, the DNA looping represses araBAD expression. But now, arabinose is present in the system, so it binds this araC dimer and causes the conformational change in the araC. This again changes the topology of the DNA by destabilizing the DNA loop. Once again, let's complete the picture and understand what happens after the loop is destabilized. You have to note here that only the N-terminal domain experiences the conformational change. The C-terminal domain, which binds the DNA, is unaffected. Since N-terminal domain is also involved in the dimerization, the entire dimer is affected when the arabinose binds at the pocket domain. And now the dimer assumes this packed configuration, such that it is forced to bind the half sites at a very short range. So there are no constraints on the adjacent site binding. So to reiterate the point, in the off state, the araC dimer can only have long distance contacts through DNA looping. But when the arabinose is bound, it changes the araC dimer configuration so that it only forms short range contacts. If you recall our point about I2 inducer site, it partially overlaps with the promoter of araBAD. Hold on to that information for a short while, and we'll come back to it. In the meantime, let's complete the picture of the operon first. At the upstream region of the inducer, you also have a full operator site. So the operator site can also have the araC dimer bound to it. And again, keep in mind that the O1L operator partially overlaps with the promoter of araC. But what about this operator 2? Which, remember, is only a half site. So the dimer cannot form a stable complex at the O2 because it doesn't have the second half site. Full sites only exist at O1 operator and the inducer. The operator 1 was caught in the loop in the off state. But now here the loop is destabilized, so the operators can engage with the araC since there's a preference for short range distance. So far, we have noticed that the DNA loop is gone and the araC dimer binds differently. But also notice that the C-terminal domain all face in the same direction, given all the half sites are oriented in the same direction. These highlighted parts of the C-terminal domain are required to activate the RNA polymerase. For instance, when the polymerase binds at the promoter of the araBAD, it is stimulated by the C-terminal domain of the araC, and it transcribes the B, A, and D genes. But in the case of operator 1, the activating region in the C-terminal domain of the araC faces away from the RNA polymerase. So at the promoter of araC, the RNA polymerase does not receive activation input from the araC dimer. araC was downregulated when the DNA was looped in the off-state condition. But when the loop is gone and the promoter is available, it is still not activated at high levels. Which means that regardless of the arabinose status, the araC is always maintained at a basal level expression. This type of regulation, where a constant amount of product is maintained, is called autoregulation. There is one more point that I haven't touched here about the operon, which is the cap binding site. At this site, the CRP-CAMP complex can bind and provide additional stimulation to the RNA polymerase, because the C-terminal domain of the alpha subunit can contact the cap site. I have discussed the details of CRP regulation in the lag operon video. You can find the link to that video down in the description. This cap site activity means that catabolite sensitivity can change the araBAD expression levels. When the arabinose is present but the glucose is also present, the araBAD is only expressed 5 times in excess. But when the arabinose is present but the glucose is absent, the expression increases 400 folds. This increase in the fold change is relative to the default state when the arabinose is not present in the cell. This also means that the import proteins araE and araF, which are originally expressed at basal level in the presence of arabinose, are also upregulated, just like the araBAD operon. and this forms a positive feedback loop. Hopefully, this entire discussion has clarified the point that araC protein can act both like a repressor and an activator. Repressor when there is no arabinose, and activator when arabinose binds to araC. And this sums up our discussion on the arabinose operon.